Yeah, no, recording in progress. Everybody wave like we do every week. Hi. Proof that you're here. Did it? Did it take it? Thanks. All right, we have a public channel, but this one's not public. So generally speaking, no, unless you know to look for it, you're not gonna find it. Okay, there we go. Now it's going to behave. All right. Let me put these over with the dish stuff because I don't want to like knock them over with my iced tea and then regret it all my life. Just say, no, that's not fun. So has anybody done the model cell lab before? Anybody ever done it before? Okay, cool. That's good. That's when a lot of like you'll see it in like the kitchen chemistry or whatever, like homeschool stuff or home science experiments at home. So that's good that that nobody's done it before because then the results will be genuinely a surprise. Woohoo! Okay, number one. These are your cups for Skittles now. The little ones are apparently too little, but some people were using like our big paper cups from inside the chest. Let's not use those. Those are for like if we have parties or if we need them for a lab, but they're too expensive to just use for Skittles every week. These little cheapy dudes, they're your Skittles. They're over on top of the machine. Got it? This is what we use. Good deal. All right, cool. Also, don't leave them on the lawn. Somebody picked them all up last week because they were scattered about the lawn. The big cups were scattered about the lawn. Let's not do that. All righty. So before we start, some of you already saw this because I put it in the forum, but some of you haven't seen it and I really wanted to share. So there's been a huge development in a story we discussed last year. So those of us who weren't with us last year, bear with me because it's still very interesting. So it was briefly discussed with the juniors, but we watched a whole documentary on this, a short documentary. But who remembers Bear Brook? Okay, remember Bear Brook? All right. And who remembers the middle child? All right. So for those of you who weren't here, Bear Brook is a very well-known cold case situation where there you look so hopeful. You look like I looked yesterday. So okay, so anyway, there were 4 bodies found in barrels in 2 pieces of first there were 2 bodies, an adult female and a child, and then several years later 2 children, all girls. They were all found in outside the state park in New Hampshire, and they were able to the reason we went over it in chemistry was because we talked about how they can use isotope testing in your teeth to tell where you grew up and in your hair to tell where you've recently lived. So they were doing that to try and help determine who these people could be. And they eventually used some forensics and found out who the killer was. And he was a guy who they call the chameleon. He's a serial killer. They call him the chameleon because he has changed his name 4 or 5 times. He goes from all over the country and he leaves a trail of destruction. He's a really not a good person. He is now dead, but he was not a good person. Terry Rasmussen, not a good person. So anyway, they eventually identified the woman and the 2 children. They had not identified the middle child. So last year, towards the end of chemistry course, we watched a video about Bear Brook case, a story of 4 unidentified bodies found in barrels in a wooded area in New Hampshire. We discuss how scientists used isotopes found in teeth and in hair to determine the areas in which a person may have lived. The technique helped narrow down the identities of 3 of the Bear Brook victims, and those identities were eventually confirmed by DNA, but the remains, dubbed the middle child, was still unidentified. Scientists knew that the murderer, serial killer, whose name is Terry Rasmussen, was the child's biological father. But that was all that was known about her. Not her name, not who her mother was, just that her own father had put her in a barrel. But now we know who she is. Her name is Ria Rasmussen. They just found out on Friday. They found her, she was on Friday, and they announced it over the weekend, which is a huge deal because this is a 50-year-old case. And they finally just solved it. Yes.
My mom saw that and she was like, oh, Miss Denny's gonna be so excited when she finds out about this.
Drew was in another room. I was in the dining room. Drew was in here.
5 times.
And I went, oh, what? I was so excited. He's like, what? What is it? I'm like, I can't believe it. This little girl's remained unidentified for 50 years, and they finally identified her just on Friday. Now, how do we do it? So here's a press release. The press release is from a guy named Jason Moon. If anybody listened to the Bear Brook podcast that I recommended, he was the host of the podcast. So it's great that this guy now gets to do the press release. Good for him. He did the work. He's earned it. It was good, right? It was real good. I listened to it again yesterday because while I was doing other things because I wanted to refresh. Yeah.
So what about the couple who like helped them?
Oh yeah, those people that were helping. Yeah.
They credited?
Yeah. No, because I'm going to tell you how they found out.
Credited?
Well, they've gotten credit so far for all the work they've done. I mean, they wouldn't have gotten this far without that. It was a brother and sister. They wouldn't have gotten this far without them. These were just like regular everyday web sleuth type people. So New Hampshire Attorney General's office says it has identified the last known victim in the high-profile cold case commonly known as the Bear Brook murders. Ria Rasmussen was just a few years old when she was murdered by her father and then discarded in a barrel near Bear Brook State Park, likely between 1978 and 1981. Yeah, it's been a long time. Previously referred to by investigators as the middle child, she was one of 4 victims found in barrels near the state park. The discovery also led investigators to a missing woman, Ria's mother, Pepper Reed, who has not been seen for decades. So what can we surmise about what happened to poor Pepper?
Dead.
Likely he did this. Yeah, it's very sad. Likely he did the same thing to her. They actually have looked at his timeline. She went missing from Houston. They know roughly when she went missing. They followed this guy's timeline for years. He was in Houston at the time she went missing. So yeah. She very likely is also a victim. Case baffled investigators since the discovery of the first barrel 40 years ago and drew, well, so 40, not 50, whatever, and drew national attention as one of the first to be solved during using a new technique called genetic genealogy, marking a turning point in how cases are now solved and investigated. What is your lab going to be about next quarter? Genetic genealogy. The same thing that they use to help identify the mom and daughters is the same way they identified her, is the same way you're going to eventually identify your suspect next quarter when we do our cold case lab. The technique was used to identify the suspected killer at first, Carrie Peter Rasmussen, and the 4 victims. First, Marlise Honeychurch and her daughters, Marie Vaughn and Sarah McWaters, and now Ria. The case has weighed on New Hampshire and the nation for decades, Attorney General John Formella said in a statement announcing the news on Sunday afternoon. With Ria Rasmussen's identification, all 4 victims now have their names back. This was the first case ever, criminal case, that used forensic investigative genetic genealogy as their way to solve it. It's not the most high profile, probably Golden State Killer is, but same thing. So in 2024, New Hampshire State Police Cold Case Unit partnered with the DNA Doe Project. If you're looking for an organization to support financially, this is a great one, which helped to identify Ria using DNA analysis and genealogical research. And look who they found in the process. In an interview with New Hampshire Public Radio ahead of the state announcement on Sunday, New Hampshire State Police Detective Sergeant Chris Elphick, so this guy Jason Moon got the interview ahead of the announcement. Good, he deserved it. That the state finally got lucky in June of this year. Investigators and the DNA Doe Project compiled a family tree with 25,000 people in it, and said that this summer they finally found a key DNA match in the family tree that led them to Ria's mother. 25,000 people. Yours has about 50 in it, I think. So yours will be much easier to navigate, but 25,000 is a lot. That's her mother, Pepper Reed. According to the DNA Doe Project, the match was a couple born in the 1870s. Next, the team says they found a 2005 obituary for one of the couple's great-great-great-granddaughters, which said she was survived by a daughter named Pepper Reed. So this is Rhea, the middle child's great-great-great-great-great-grandparents that they went all the way, traced all the way back to in order to give her a name back. Team could find no records for Reed after the 1970s. Gee, I wonder why. But the DNA Doe Project says once they had her name, they found a birth record for her daughter within 30 minutes. It took them 18 months to piece together 25,000 people to get her mother. And once they had her mother, they had her in 30 minutes. They had her in 30 minutes because they were able to search her mother's name with birth records, and they found her in California. Had they just randomly searched for Rasmussen birth records, there are thousands and thousands of people with the last name Rasmussen. Plus, that guy changed his name every other week. So it would have been very difficult for them to just search birth records for a random Rasmussen and then try and trace every one of those kids to see if they're still alive, if they're missing, if they're whatever. That would have been nuts.
Did they find Pepper Reed's body?
No, we don't know where Pepper is. Pepper is one of many that Terry Rasmussen probably left behind that we will not find, or we will not find anytime soon. To figure out the identity of Jane Doe, that's Rhea, right? We had to find her mother. And DNA Doe Project's Matthew Waterfield said in a press release, it took us almost 18 months to identify Pepper Reed. And once we knew her name, it led us right to her daughter. So from there, Elphick and DNA Doe Project staff tracked down Rhea's birth certificate, and through that, they found and DNA tested another family member, and they officially confirmed her identity on Friday, September 5th. So middle child has been named, finally, after 40, almost 50 years. That's just crazy. Yeah. So it's a very big deal. And if you remember, I believe it was discussed in the documentary, but one of the actually the lead pioneer in forensic investigative genetic genealogy is Barbara Ray Venter. And she has a separate company on her own that does this now. And they were part of this as well. And she was the one who did the first ever criminal FIG case, which was finding out who Terry Rasmussen was and the other girls were. And she was also part of this. So it's full circle. Are you very happy? I was like that yesterday. I spent all yesterday going, oh, people are like, why do you get so attached? I get attached. It's a child. She should have had her name all along. She was a toddler, for goodness sakes. So this is awesome. This is really, really, really awesome. And this is kind of like what you'll be doing. You'll be doing forensic investigative genetic genealogy. And here's a real case where a real person was just now found. Poor Ridley. That's how I was yesterday.
This was like one of the first cold cases I ever learned about.
Yeah.
So this is a big deal.
It's a very big deal. It was a nationally recognized case where people were spending forever trying to figure out who this guy was. And he got caught. For those of you who followed this story, he got caught because he killed his live-in girlfriend / wife. I don't think they were really even officially married, but her friends were looking for her and they couldn't find her, and he kept making excuses for her. And do you know how they found her? Does anybody know how they found her? Do you remember from the podcast? They found her in like a basement area off his garage, buried under 3 feet of kitty litter.
Oh yeah.
Mm-hmm. Yeah. And then when they brought him in for questioning about all of that, they said, do you mind if we get your fingerprints? Because he was going by a different name. And they got his fingerprints, and then they came back and they're like, that's not your name. You have another name. And this is who you are. But that wasn't who he was either, because he had a series of names. The reason his fingerprints were in the police system is because he had gotten arrested for child abandonment, because he was at a campground in the early 80s or 70s, late 70s. Lisa. He had a little girl with him, and he told this other couple that was camping near him at the campground, this is my daughter. My wife died of cancer. I'm just overwhelmed. And they were like, well, our daughter can't have children. We'll take her. And it was like the early 80s. So they were like, sure, let's just adopt your kid from the campground, because things were weird. And it was the 80s. And they took her home, and then she started telling stories about things that made them think she had been abused. And they looked into that. And eventually, over time, she started to question if she really was his daughter. She was not. She was not his daughter either.
You are not the
He
father.
basically his MO was to go through the world and either find a woman with children, or have children with a woman, and then get rid of the woman and use that child as a lure to bring in the next woman. And he did it again and again and again. And he probably has a trail of people we don't even know about yet. But it's incredible that we just talked about this just a few months ago. And I was saying, one day hopefully we'll find out who she is. Now we know. So that was very good news. I was very excited. I was very excited. Yep. So let's move into today's lesson now that you have your very good news. That's your what do they call it? Segment of hope for the day. So let's talk about how cells got discovered in the first place. Now I'm going to be handing this to you in a little bit. It's a cell you can color and label while we go through lesson. Not yet though, just so you know. All right, so that's all right. First, we have this dude, Robert Hooke. Okay, so 1665. He looks kind of sick. Probably everybody was in 1665. And he looked at cells under a microscope, and he was looking at cork. And he saw that they looked like these little chambers. And he's the one that came up with the word cell. So yeah, prison cells were not named after cells. Cells were named after prison cells. Pretty much. They look like little cells where you might stick somebody forever and ever, like Terry Rasmussen, which is good. Yeah, he was there for his forever and ever. So yeah. It's one of those like, I'm not sad he's gone, but the information that he had that he took with him kills me. So anyway. Yeah. Well, cancer killed him, and rapidly and badly, apparently. Yeah, really awful. I wouldn't wish that on anybody, but it's kind of like I'm not too sad in this game. I'm terrible. Anyway, so then we have Antoine. Anybody want to try his name?
Oh,
Do you know it?
Antoine van Leeuwenhoek.
Leeuwenhoek? Luffenhoek. Antoine van Luffenhoek. Yep. Okay. So he was the first one to see living cells, living organisms in a drop of water. So you wonder, what are these dudes in the late 1600s using for microscopes? Like, if you've ever noticed, there's a little lamp over here made out of a microscope, and that is a like 1890s to 1910 style microscope, and boy, is that thing janky compared to the ones we use. So what were these people using? Well, they look like this. So Van Leeuwenhoek's microscope is this weird contraption over here. Yeah. So you can see, like, you put the little sample there, and then there's like, you pull it up and you peer through it like that. And Hooke's microscope, I think this is ingenious. You need good light, right? When you turn on your microscope, it's like you have to turn the light down sometimes with the diaphragm because it's almost blinding. So how do you get the light? An oil lamp, and then water act like a magnifying glass, amplified that light right down onto the platform that he was using to bounce it back into the so he could see when he looked down through this little thing. And it looks basically, it's just a very small telescope. A very small telescope with cool light.
I like that.
I love this. This is so steampunk. I want that in my life. It's really cool. Right? Very cool. So after those two guys in the 1600s, we move into the 1800s now, and we have Matthias Schleiden. And he looked and said, oh, all plants are made of cells. This is one of those things that we were talking about spontaneous generation with the juniors this morning. Some things are real obvious to us these days because that's all we've ever known, but they were not obvious to people before. So you have to make these statements in science. All plants are made of cells. I have now made a statement, and it is now science. Like, that's kind of how it works. Like, it took some guys back in the day to prove that if you put a steak on the counter and in 2 days you have maggots, the maggots did not come from the steak. The steak did not generate the maggots. Like, they thought that it did. So it took them using experiments to prove that the steak did not generate maggots, that the steak's not alive anymore, so it cannot produce life, that life comes from life. So yeah, this is one of these obvious ones to us. All plants are made of cells. Oh, big deal. It was a big deal in 1838. And then you get Theodor Schwann, and Theodor Schwann said all animals are made of cells. Oh, thank you. Now, he also invented the first rebreather. You're not ready for this. I'll just tell you before I show you the picture, you're not ready for this. Okay, the first rebreather, right? Like, you put it on, the air goes through, it gets filtered somehow, and then you breathe it again, so you're not breathing external air, right? In 1839, You're not ready. I promise. It look like this. Yeah. All I can imagine is sorry. Looking at this picture of him in his rebreather yesterday, and I was imagining him sauntering up to a lady and being like, "Hey, I'm a scientist and I built a rebreather." Her being like, "Yeah, show me." And then him coming out in that.
It looks like a really weird trombone.
So suave, man. It's like, and this picture's the best one. Like, what is this?
Scuba gear.
No. Way to go, Theodore. You had it going on with your rebreather. You man, you. Apparently it worked. I'm not even sure how exactly it works, but I think the center strap is particularly fascinating. Like, divides his head into this
I
way.
didn't even notice that. Yeah.
It's classy, isn't it? I mean, that's pretty awesome. So hooray for people who are inventive and creative, because that's as bizarre as it is, it's still pretty darn cool. I didn't invent a rebreather. What am I laughing at? He just looks ridiculous. But told you weren't ready. All right. And then we get to Rudolf Virchow, so 1855. And he said, all cells come from existing cells. Okay, another bold statement that now we go, well, yeah, of course. But in 1855, people are like, really? Oh, wow, that makes sense. Okay. So these are profound statements made in their time and day. So if you've got your phone, we can play now.
Oh, my one time I forget it. Date.
Yeah, I love little food.
One time I don't remember.
So we're gonna do our cell theory, guys, and we're gonna ask questions about them, huh? You don't need it. It's just for fun.
That one makes it sad that I didn't.
All righty. So, oh wait, you need it, Jordan? There you go.
Right, okay.
Got it?
Wait, no.
Nope.
Why? There we go. Okay.
There you go. All right. I don't know why it's giving me trouble. It gave me trouble the other day, made me click something every time. Okay, so our first question is maybe, come on, load it up. Why are we being so painfully slow? Here we go. It's coming. It's just being slow because it's the first time it's been in today. Loading. Thank you. We know you're loading. I know. Isn't that great? Goodness. It's spinning. Look, it's spinning. Wow, it's very slow. It should be faster on the subsequent slides. It's just that first one. All right. Okay. Start Menti. Here we are. First question is, here we go. We are going to rank them in the order in which they were born. First at the top, last at the bottom. I'll give you guys a minute to do your ranking. There we go.
Robert.
All right, so let's see our answer. Oop, there goes one more. Oh, and again, no, they're still moving. All right, so in what order were they born? We had Antoine, Robert, Matthias, Theodore, and Rudolph in the following order. These two are pretty darn close in age, and these two are pretty darn close in age. Matthias Jakob Schleiden. All right, good name. All right, so next one. I don't know why it's so slow today. It gave me trouble when I reviewed it too, and I'm like, why are you doing that? Usually it's instantaneous, but go. Okay, our next question is, if it will show me the question sometime today, maybe. Okay, well, you're getting answers even though the question hasn't shown up. Where was Robert Hooke from? Why is it answering it that way? There we go. Dutch, English, Polish, or German? Polish, you think? Dutch, English, most people seem to think he's Polish. Robert Hooke was English. Ah. He looked so sad. Had that very serious English look about him. All right, our next question is. Yeah, I don't know why it's doing this today. I can't make it not do it, so. Should not have to hit start Menti on every one of them. Where was Antoine van Leeuwenhoek from? Polish, German, English, or Dutch?
None of them are Polish.
None of them are Polish.
I wouldn't think anyone else.
Nobody considered this little part of his name a big clue. 3 of you did.
German.
He's Dutch. Dutch.
Wasn't German found as Dutch? He wasn't.
No. Like country. No. It's a different country and different language. But very similar, but different. Okay. Yeah, the Netherlands, exactly. All right, sorry, next one. I don't know why these are, I hate that they're slow today. It's making me crazy. It's gonna ask where our next dude is from. Where is Matthias Jakob Schleiden from? Jakob Schleiden? Matthias Jakob Schleiden? Gee, nobody thinks he's English with a name like Matthias Jakob Schleiden? That's the most English name I've ever heard. Jakob Schleiden. So I laugh at every time I see the name Jacob in any form, I have to laugh. Because years ago, my husband was typing up a list of guests, customers that he was like inviting to an event. And he was going back and forth between reading and typing. So he asked my older son to read. He's like, can you just read the names to me and I'll type them? So Cole was reading the names, but a lot of them are names from different cultures, like a lot of them, because his company's Nokia. Nokia's Finnish. So there's that. And then there were a lot of Asian names. So he was doing his best to pronounce all these names. And you get to this one person, he's like, Jacob something or other. And Jason was like, who? And he's like, Jacob Williams. He's like, it's Jacob. That many like weird names that it came out Jacob, and we were like, that's great, man. So he was German, obviously. Jakob Schleiden, Matthias Jakob Schleiden is going to be German. Yas. Well, then we got Rudolph, right? We got our man Rudolph Virchow. I'll give you a chance to answer that one. You should be able to move ahead even if it hasn't popped up here yet. Oh, Schwan. Yeah, Schwan is next. Sorry, not Virchow. Schwan. Theodor Schwan. Dutch, English, Polish, or German?
This might actually have a chance to be Poland.
This one might have a chance to be Polish. So Schwan is also German.
What?
Yup, Schwan. Theodor Schwan. So lastly, we have our Rudolph Virchow. Why they're being so crazy. Do. Come on, pop the little button up for me, please. So slow today. So where was Rudolph Virchow from? Is he Dutch, English, Polish, or German? Everybody's like, hey, we think we figured this guy out, huh? He's the last, so he's got to be the Polish guy because I put Poland in there. It could have just been a wild card. There might not be anybody who's Polish. He's Polish. So yeah, there you go. All right. Rudolph Virchow, very Polish. All right, how about this next one? What you got?
And here it says discoveries by German scientists. That guy Sheldon or Schwann and Virchow.
Yep, and he's Polish. I think he may have been living in Germany at the time. I could be wrong, but he's
What
Polish.
time period?
We did it. It was 18 something.
2018.
Yeah.
That's Germany,
Mhm.
history book.
Yeah. Well, it was Prussia, right? So you had all the Prussian influence everywhere. All right, so our next question is, if it will come, I don't know why this is doing this to me. It's usually like this, right?
And you snapped and it happened.
Yeah, it did. Who's known as the father of microbiology? It was what?
Just answer that word first thing.
Oh, did it? Well, that's weird. It shouldn't have done that.
Get the answer, it just switched before I.
It's weird. I wonder why it's out of order like that. Well, the father of microbiology is our buddy Antoine. Antoine. And then we have uh-oh. Uh-oh. There it goes. I'm like, where's my spinny square? There it goes. Spinny square. Got a lot of choices. Our little dudes again, and it's asking, well, who has a type of nerve cell named after him and also coined the term metabolism? Virchow,
Wow.
Schleiden, Schwann, Leffenhook. Hook.
He's using AI.
He's using AI.
Like,
Cheater. 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 Jakob Schleiden.
Okay.
So there are Schwann cells in your nervous system. Very type of nervous cell is a Schwann cell.
Animal.
Very good. Yes. There you go. Okay. It's just like I don't have any more. That's the best I can do right now. This is painful. It's not the Wi-Fi. It's the actual software on their side. Who is the father of modern pathology? What's pathology? Do you know? Yeah, disease, study of disease. A forensic pathologist is somebody who's going to give you an autopsy. Should you ever need one day. Your medical examiner will likely be, medical examiner is your county or city job title, but your field of study is forensic pathology. Coroners and medical examiners are different. So coroners do not have to be medical doctors, but they do provide a death certificate. A medical examiner does an autopsy. So you can have both in your county, depending on how it is. Actually, I have a friend who's a funeral director who ran for coroner of Forsyth County several years ago. And I believe our current coroner used to be a funeral director as well. Yeah. So our father of modern pathology is Rudolph Virchow. Yay, Rudolph. Flash. No, I don't get it. It never does this. And today it's like trying all of our patience, but that's okay. We're patient people, right?
I'm not.
You know how to wait.
So.
I'm good. I take my meds so I can wait a minute. There you go. I had a coffee, so I have more patience today. All right, who was also an astronomer, geologist, meteorologist, and an architect? What do we call somebody who is lots of different things? Polymath. Who was the polymath here? Schleiden. Rudolph. Van Leeuwenhoek. So our polymath was Robert Hooke, our earliest guy, way back in the day. I think I'll build a building and look at cells under a weird microscope using an oil lamp and a bulb of water, because I'm all kinds of fun things. Oh, and by the way, I'll tell you about the weather. Give me another famous polymath. What you got? Leonardo da Vinci? What you got?
Galileo.
Galileo? Could be. Give me an American polymath. You got one?
Me.
You? Anybody got a good American polymath? Thomas Jefferson, Benjamin Franklin, both polymaths.
What about Nikola Tesla? Edison.
Tesla, yeah, he eventually was an American, but mostly focused in one area, but still very, very bright. Started in Serbia, right? He's Serbian born.
Oh, what was his name? Oh, dang, what is his name?
What'd he do?
Nazi rocket scientist.
Oppenheimer?
No, not Oppenheimer.
Oh, vinegar?
Yeah.
No, not vinegar. Not him. Hold on. Werner von Braun. Von Braun. Yeah. Werner von Braun. Aristotle. Okay, yeah. This one doesn't want to play. Oh gosh, there we go. Jeez Louise. Sid the Science Kid. Okay, there we go. We got a couple. All right,
My
yeah.
dad walked into my grandma's senior living, and one of the older ladies who has dementia walked up to him and said, and
Bill
and
Nye?
the thing is, he dresses up every day to like, she likes being really presentable, and she just like walks up and just like, Bill Nye, is that you?
Is that you?
And he just went back to the car crying.
So who, though not Jewish, fought against anti-Semitism? Mm-hmm.
Which one was German? Like, which ones were German?
Schwann and Schleiden.
Okay, so not them.
Not necessarily.
Anti-Semitism is anti-Jewish sentiment, if you're wondering what anti-Semitism means.
It's probably Robert.
Oh, and look at the shifting around. It was our friend Matthias Jakob Schleiden.
Yes.
Now, last year, remember we talked in chemistry, we talked a lot about nuclear science and stuff. We discussed how there were a lot of those guys that were kind of coming of age in their career, like right during World War II, and had to make decisions. Like some of them fled and got away from the Nazis. Others were part of that organization. And then there were others who like specifically, do you remember that it was Schrodinger and Heisenberg. It was Heisenberg who thought this would all be over really fast. He just wanted to keep his job and he kept a low profile. And he's like, if I just shut up and do my job, this can't last long. Like nothing really bad's going to happen. I'll just keep my head down. Right. All right. So who lived to be oldest? What do we think? Rudolph?
I'm gonna say Robert. They had good health in England.
Always the carrot.
But,
Hey, somebody found a carrot emoji.
Poland.
Poland. All right.
Remember that being up.
Not so much, but hey, it's always fun.
The country Poland did live longer than all of them.
I was gonna say this is accurate, actually. Sliding into your sliding into your DMs. I love it. Sliding into your DMs. That's great. Yeah, the most amazing people. This is why I love this. Hey, we got a lot of sliding into your DMs. All right. Hold on.
And it's so close to it. I just made it up.
That's great. So here we go. Here's Hook, 67. Here's Schwann, 71. Schleiden, 77. Virchow, 80. Van Leeuwenhoek lived to be 90 in the 1600s. That is like ancient. That is ancient in the 1600s.
And
Ancient. But hey, that's crazy. Nobody lived that long. That's impressive. All right, so now we're going to talk cell theory. So the main parts of cell theory are all living things are composed of cells. Cells are the basic units of structure and function in living things, and new cells are produced from existing cells. These are our main points in cell theory. So we'll talk about types of cells. We have prokaryotes and eukaryotes. Somebody tell me the big difference between prokaryotes and eukaryotes. Who knows? Eunice, do you know?
eukaryotes are cells that enclose their DNA nuclei, and then Prokaryotes are cells that do not.
There you go. Your big difference is that your prokaryotes lack a membrane-bound nucleus. And I screw that up every time I try and say membrane-bound nucleus. I don't know why I have trouble with that, but I do. And then your eukaryotes have a membrane-bound nucleus. Are you a prokaryote or a eukaryote?
Bro,
I like dining.
Who votes for being a prokaryote? Who votes for being a eukaryote? Yes, you are a eukaryote. I am a eukaryote. We are all eukaryotes. That's how you are. You are a eukaryote. So your eukaryotes have a membrane-bound nucleus and organelles that are organized and whatnot, whereas prokaryotes are a little more primitive. They have their DNA in a plasmid form and it just kind of wiggles around in their cytoplasm. It is not bound within a nucleus within the cell. So it is less highly evolved. All right. I want you to take a look at these dudes for a minute. These are all very tiny little microorganisms, and you're going to try and decide mentally where they're going to go. Yes, Lucas.
Is there an advantage to having DNA in the nucleus as opposed to not?
It just is, I mean, yes, because I'm not sure how to frame it. It's a sign of higher level of organization. So, like, the more organized and complex you are, kind of in many ways, the more advantages you're going to have. I'm not sure specifically how you could tag it. I'll look and see if there's like a nice brief answer as to why that is. But all higher organisms are going to have a membrane-bound nucleus. You're going to keep your DNA nice and protected within that nuclear membrane inside of your cell itself, which is, it's going to allow it to be better protected for one thing. That would be one easy advantage. All right, have you kind of decided in your head which way these suckers are going to go? What's going to be a prokaryote versus a eukaryote? Let's see where they go. Yeah, that's pretty primitive looking. Oh, paramecium. Okay, yeah.
the sausage links.
Sausage links. Yeah. Well, where's the last guy going? There we go. So if you notice, those are all pretty basic, right? And these are a little more complex looking in general. These are typically the types of bacteria that you'll hear them described as cocci. Those are like the little round ones, rods. And then some will be referred to as spiral, and that has to do with their shape. And they will, sometimes the type of like antibiotic you take depends on the type of bacteria, meaning like kind of how it's shaped in a way. So cell structure, by the way, I'm wearing a cell today.
Yay.
One of my favorites. One of my favorites. So I'm going to hand you some blank diagrams to color as we label. Color and label as we go. I'm going to give 2 over here. These are you 2. 1, 2, 3, 4, 5, 6, 7. Pass those in that.
No, no. No. It's allowed to nowhere else. Not just in this.
Let's see.
Thanks. Okay. You can just kind of label them as we go. You can color them if you have your colored pencils with you.
Benny,
Something to do while we talk. Yes.
why are there 2 copies? One is and then
One is plant and one is animal.
See, it says on the top. No.
Plant and animal. So you got plant on one side, animal on the other side. And you can keep this. You're not giving it back to me, so. So we're going to start by talking about our basic structures and our basic organization. Remember how I was telling you about how cells have that phospholipid bilayer for their plasma membrane, their cellular membrane? There's a good diagram of it. Got your little hydrophobic heads and your hydrophilic tails. Remember, hydrophobic means water hating, hydrophilic means water loving. Right on. So we have a cell membrane. Anybody want to tell me what a cell membrane is? Cell membrane or plasma membrane, Eunice? What you got?
I can't find it.
Can't find it? Okay, Caitlin, what you got?
It's a thin, flexible barrier called a cell membrane.
Mm-hmm.
Yeah.
Barrier is good way to put it. Yep. So it's a flexible barrier around the cell, protects the cell. So that is your cell membrane. It is present in all cells, even your little unicellular organisms to the cells of your multicellular organism, prokaryotes, eukaryotes. Everybody has a cell membrane. The phospholipid bilayer is a universal component of all cell membranes. Its role is critical because its structural components provide the barrier that marks the boundaries of the cell. Structure is called a lipid bilayer because it's composed of two layers of fat cells organized in two sheets. There's one, there's the other. And here's their little tails that come in. So they have heads that are lipids, and this little bit in the middle is going to be hydrophilic. And it is selectively permeable, just like we're modeling in lab. So now we will talk about cytoplasm. What is cytoplasm? Somebody, anybody? All right, Eunice, you got this one?
It's a portion of the cell outside the nucleus.
Yeah, so your cytoplasm is your material inside the membrane, not including the nucleus. It's this jelly-like substance that everything is floating around in. All cells have cytoplasm. Oop, did I go too fast? There. Present in all cells. All cells have cytoplasm. All right, so our next one is our cell wall. Now, what's a cell wall? Anybody know what a cell wall is? Eunice, you got that again?
So the supporter shape that protects the cell.
Yeah, okay, so your cell wall is found in many organisms, including plants, algae, fungi, and nearly all prokaryotes. So your prokaryotes, which are your non-membrane bound little critters, they have cell walls. Do animal cells have cell walls? No, right? And you can see that on your diagram. Your animal cell does not have a cell wall. Interestingly, the cell wall of plants and of a lot of smaller prokaryote type critters is composed of cellulose, which we call fiber. That's why you can't digest it. Actually, that's what your fiber is. It's the cell walls of all your different vegetables and plants that you're eating. But in mushrooms or fungi, it's made of chitin. And interestingly enough, chitin in nature is also the same compound that creates the exoskeletons of arthropods, like bugs and whatnot. Same compound, chitin. So it's not found in animal cells. It lies outside the cell membrane, and its main function is to provide support and protection for the cell. Eunice?
Are cell walls only for prokaryotes?
No, plants, algae, fungi, and nearly all prokaryotes will have cell walls, but not
a single nucleus.
not a what?
So. Oh, okay.
Got it? Okay. Yep. All right, and again, composed of cellulose in most of those organisms. All right, so what's a nucleus? Okay, Yusuf, what you got? There you go. So yes, it's in almost all eukaryotic cells, including plants and animals, and they have a nucleus. And the nucleus, prokaryotes do not have a nucleus, right? And it controls most cell processes and contains your DNA. The nucleolus. Anybody know what that is? Your nucleolus is inside the nucleus, and it's where your ribosomes are made. And then your nuclear envelope is a double membrane layer around the nucleus. So that's the nuclear membrane, is your nuclear envelope. You can call it a nuclear envelope as well as a nuclear membrane. And it's a double membrane too, just like your cell membrane is. And it allows material to move in and out of the nucleus. Remember, it's nucleus, not nuclear, not nucleus. We don't have nuclear war, nuclear weapons. It's all nuclear, 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 right? Same word again. So organelles, by the way, means little organs. Oh, they could have called them organettes too. Yeah. Organets would have been more fun, I think.
All right, I'm going to try and pick up where we had to leave off because of tech problems. So I'm going to make this quick. So we were still in organelles. We were talking about how organelles are little organs, and we were talking about vacuoles in particular. We discussed how the organelles involved in storage, cleanup, and support are vacuoles, which include a couple different types, but in general we're talking about a space within the cytoplasm of a cell closed by the membrane, typically containing fluid, and these can help sequester items or maintain a fluid balance. A central vacuole is in a plant, and that helps for maintaining water, which maintains the plant's structure. Contractile vacuoles are inside of a lot of protists. They're used to pump water in and out. They can also be used in locomotion to some degree. And then we have food vacuoles, and these are for storage of molecules and food sources for the cell through phagocytosis. Phagocytosis is the process of the little vacuole kind of turning itself into like a Pac-Man shape and wrapping around whatever it wants to capture, and then sealing itself back off, causing a little bubble of whatever it captured to be caught with it inside itself. That process also goes on with certain organisms, like amoebas, for example, can use their cell membrane to undergo phagocytosis and eat food from outside of their bodies. And then lysosomes. We talked about lysosomes. These are organelles in the cytoplasm that have degenerative enzymes in them. So basically, lyse means to break. So a lysosome contains the chemicals that are used to break things apart or break down things. They're used to they're like the cleanup crew. They destroy, I can't speak, I've been talking all day, I'm sorry. They destroy things so that they can be kind of cleaned up and disposed of by the cell. Next, we talked about cytoskeleton, which is the structure of the skull. It's made of little microtubules and filaments. And a centriole is this barrel-shaped organelle over here to the right, which is involved in helping produce that cytoskeleton and, well, produce the structure of the cell. And then let's see, we moved on to protein builders. So we talked about endoplasmic reticulum, which is this kind of wiggly stuff outside the nucleus over here. Endoplasmic reticulum, we lump it into two main categories, a rough endoplasmic reticulum and a smooth endoplasmic reticulum. And you will find ribosomes on your rough endoplasmic reticulum. These are involved in protein synthesis, and your ribosomes in particular are where your RNA is taking the information in your DNA to the ribosome by RNA, and then it is transcribing that information into proteins. We also have Golgi apparatus or Golgi complex. They look like a little stack of pancakes, and they function as a factory in which proteins received from the endoplasmic reticulum are further processed and sorted for transport to their eventual destination. Next, we have energy. So chloroplasts in our autotrophs take energy from the sun and convert it into stored energy in the form of sugar. Oh shoot, there's an alarm, sorry. Involving the chloroplasts, involving the chlorophyll within the chloroplasts. And we're going to watch a video in a couple weeks where it kind of takes you like inside of a chloroplast. It's really kind of cool and shows you around all these different structures. You also have mitochondria, and these are in your eukaryotic cells for the most part, and this is what you call the powerhouse of the cell. This is where your ATP is going to be generated, and ATP is your energy molecule. Excuse me. I'm not going to click these two links right now, but I'm going to put them in the resources and in the weekly email. These are two links where they've taken the organelles in a cell and made an analogy to the TV show The Office, and it's a good memory device if you're trying to remember what all these different organelles do. It's a good way to go about kind of helping you memorize it. All right, we talked about transport. So we have a phospholipid bilayer in our cell membrane, as you know, and it's semi-permeable. It's also called a plasma membrane. and it's selectively permeable, so only certain things can pass through, other things cannot. So you have active transport. This requires energy from the cell to move materials in and out, while passive transport can be done without using cellular energy. And so these are things that can move in and out of the cell without requiring energy. And the method used by a cell to pass molecules in or out is determined by the size of the molecule as well as the concentration of the molecules on the inside versus the outside of the cell. Most membranes are covered with tiny protein channels, and we'll talk about these a little bit. And these act as gateways or pathways for things to move in and out. If a molecule is tiny enough to fit through a special protein channel in the plasma membrane, it will use a form of active or passive transport to move through. If the molecule is too large to fit through the protein, it will have to enter through a vesicle. A vesicle can be thought of like a tiny pouch that surrounds the molecule and ushers it through the opening of a cell membrane. If you hear birds tweeting, it's because I'm outside. It's a nice day. And there are people yelling across the river, and I don't know why. So we have passive transport in the form of simple diffusion. These are things that are small and nonpolar, and they can pass across the plasma membrane. Facilitated diffusion is going to require energy, and these are larger molecules that have to go through particular channels to get through. Excuse me. So we talk about osmosis, and this is the movement of water. Osmotic pressure is the pressure that must be applied to the solution side to stop fluid movement when a semipermeable membrane separates a solution from pure water. So if you look, you'll see that you have hypertonic, which is a higher solute concentration outside the cell than inside the cell. And if you look at the graphic to the right, you can see that cell appears to be almost crushed because there's a higher concentration outside than inside. And you have hypotonic, and in that case, the solution outside has a lower concentration than the solution inside. So that cell looks like it's going to almost explode. It's very swollen. Isotonic is what you want. That means it's at the proper amount of pressure, osmotic pressure. And cytolysis, lysis again, lyse means to break. So cytolysis is like your cell is being broken and all your cytoplasm and all of its innards are being spilled out. So isotonic is the same on both sides. Excuse me. And then we have aquaporins. Aquaporins are like these little channels that water can move in and out of. And they are integral membrane proteins. They serve as channels that transfer water, and in some cases, even small solutes across the membrane. So transport proteins are proteins that transport substances across biological membranes, and they can be found within the membrane itself. Some of them form channels, some of them are gates. So let's have a look. So you have channel proteins. That's like the one at the top, and the green one there. So this is like a pore. It's like a tube. Water or other things can go in and out just by moving through the little tube or the tunnel. Gated proteins are exactly that. They have little chemical gates that open and close and allow things to either pass through or not pass through. And then you have carrier proteins. And these are for specific molecules, ions, or substances. So they will only let certain things through. They're similar to a gate in that they open and close, but they're very specific as to what they let through. And it's almost like it actively moves through, you can tell in that picture at the bottom, the green one, that it's those little particles, whatever they're representing, are actually sort of being like moved in and then moved out as if it's actively being carried through the channel. Just another graphic. So homeostasis in cells. If you remember, homeostasis is when you have a balanced situation inside, outside the cell. We all need homeostasis to function. So when we talk about unicellular organisms, the three we're dealing with here, at the top is your paramecium, on the bottom left is amoeba, and the bottom right is a euglena, and we will be looking at these in just a couple weeks. So of your unicellular organisms, in terms of their numbers, unicellular organisms dominate life on Earth. They include both prokaryotes and eukaryotes. Prokaryotes, especially bacteria, are remarkably adaptable and live almost everywhere in the soil, the leaves, the body, the air, even in your body. Sorry, ocean and air, I said that wrong. Eukaryotic algae are found in oceans, lakes, and streams around the world. Yeasts or unicellular fungi are also widespread. Yeasts play an important role in breaking down complex nutrients, which makes them available for other organisms. So when we talk about how life is organized, it goes from atom which joins with other atoms and makes a molecule. In this case, we can say molecule because we're talking about living things. So compounds would be appropriate as well, but molecules would be, compounds would be inclusive, but molecules would be appropriate in this case because we are talking about organic things, living things. So atoms join together, they form molecules. Molecules join together and form cells. Cells join together and form specific tissues. Those tissues form organs. The organs join together and form systems, and the systems form an organism. Here are the results from your homework. And you can pause that and check your answers. So again, here's our organization of life. We're going to start with hydrogen as our example here. So we have atoms. Say you have a hydrogen atom. It can join with other atoms, and now it's water. That can join with other molecules and form a cell. In this case, we'll say an epidermal cell, which is a type of skin cell. And then the tissue it's going to form would be skin, particularly epidermis. And then the organ that it would be a part of is also the skin. And then that would be part of the integumentary system, and then you get an organism at the end. So again, atoms to molecules, molecules to cells, cells to tissues, tissues to organs, organs to systems, systems to organisms. And then I passed out some coloring sheets in class today that you could label and color while I spoke. This was the final result for a plant cell and the final result for an animal cell.